Hi everybody. I'm going to demonstrate today some measurements we can take from living bromeliads that don't harm the plants and allow us to track their growth and development over time and compile data sets by many different scientists, many different researchers for lots of plant individuals. But before I do that, I kind of want to walk you through a bromeliad's growth and development so you can understand the parts that we're measuring. Okay, bromeliads and many other monocot plants have a body form called a rosette. So all plants share this sort of modular body where we make a leaf, then stem, then leaf, then stem. In the case of a rosette plant like this, the stem is minimized, so it looks like it's all leaves kind of coming out from one point. But in fact, there is a little bit of stem between every single leaf. And also, when the leaves are made, they're made in a spiral. Who's making the leaves? Well, at the center of every single rosette, you go down, down, tiny leaf, tiny leaf, tiny leaf, tiny leaf. We probably can't even see it, but there is, at the very core of that, a big kind of balloon of a bunch of cells called the shoot apical meristem. And the shoot apical meristem is dividing out, making leaf, and then another leaf, or like leaf stem, on and on and on to a certain point. Now, in response to perhaps the plant's age, its perception of the environment, we don't know in many cases, in most species of bromeliads, eventually the shoot apical meristem is going to change, and instead of making more leaves, it is going to make a flower stalk called an inflorescence. So that's initially going to have some kind of short, often different looking leaves, a much, much longer stem, and eventually individual flowers. So these flowers haven't opened yet, but this whole structure is an inflorescence. So the shoot apical meristem, deep down in there, changed and then produced this entire stalk. And that is going to be the end of this entire rosette. I also call that a parental ramet's life. Once those flowers open, hopefully are pollinated somehow and produce seeds, that's kind of it for this entire structure. However, most bromeliads have an additional way of continuing their life before or after that big event. Every single leaf in the rosette, and this is the true for all plants, every single leaf has a bud right above the leaf's attachment to that stem. And in this case, it's deep down in the core of the rosette. So every single leaf has a little bud deep down in there. Now, for the most part, these are quiet and not really doing anything. But again, in response to, we don't really know, light, time of year, what else is going on in the plant. Some of these kind of wake up and start dividing and produce a new little rosette. These are called axillary ramets or pups. This is a genetic clone of the big parental ramet here. They are connected at this point, and probably there's exchange of materials happening there, but we don't actually know much about that. Am I providing any sugar this way or this way, water? We really don't know. But in this case, here's an axillary ramet, here's another little one, etc., etc. So what's going to happen over time is that this main parental ramet is going to die and kind of fall apart. But these will then continue on and form an entirely new kind of similar sized body with its own inflorescence, and it goes on and on and on that way. Okay, so that's general bromeliad development. So now we want to talk about what are some data points we can take to track over time how these bodies are growing and developing. Well, if you just want to know how big something is, right, you could weigh it or mass it on a balance or something like that. But it's going to be a little tricky if we wanted to compare these two plants, because much of the weight, and I can feel it as I'm holding it, is coming from these pots and from the soil that they're in. and that's holding a lot of water. So it could be the case that this was much more recently watered than this one. It's going to weigh a lot more, which would make us kind of in error think this is a much bigger plant, when really it's just water weight. So when you're weighing plants that are alive, just be aware of kind of variation that might not be so meaningful like that. So we developed some other ways of tracking and measuring how these plants grow over time. So I'm going to teach you two sort of quantitative or numerical values we can take on plants at different intervals of time, and then what to just check for with those meristems periodically as well. So I'm going to show you longest leaf length and number of chlorophyllous leaves. That's just a fancy way of saying the number of green leaves. So bromeliads can build their bodies and make them bigger by adding more leaves and lengthening leaves. So we're going to kind of capture that here with these plants. Okay, so to find the longest leaf, you need a flexible piece of wire and a ruler. And again, look at past day sheets or just sort of decide as a team or a class what sort of units are we going to use for measurement so we can compare across data sets, and write down what you use. I would recommend using millimeters. Okay, so the first thing we're going to do is with our eyes, we're going to try to identify which leaf do we suspect is the longest leaf length. Okay, and I'm pretty good at this. I often can get it at first pass. Okay, I think we've got a real contender here. So what I do is I take my wire and I push it along the top of that leaf and down into the tank. And there's sort of like a point at which I can feel it hitting the tissue inside there. Now I could keep pushing, but I'd be punching through tissue. So this is sort of a gentle touch to figure out we don't want to harm these plants by pushing too far into that. Okay, so once I feel like I have it where I want it to stop, I run along to the very tip and pinch it with my fingertips. And I'm like, this is the length of this leaf, who I suspect is the longest leaf on this plant at this time. So I want to see, are there any leaves that are longer? So just keeping that pinch, I come over here, and that's right at the same, so not longer, so that one is not quite bigger. And yep, that's a little bit shorter than this one. Here I'm kind of keeping an eye on it. Much shorter. And a little bit shorter. Yeah, sometimes they're like really close. So once you feel like you've kind of checked it out, you're like, okay, yep, that was the one. So again, with that firm pinch, running along on my ruler to 170 millimeters. And I have these little twist ties. You could use a hair tie or a rubber band or something like that. I'm just going to use that to mark who is that leaf, and I might record it in my notebook. 170 millimeters is the longest leaf on this individual on this day. And I'd keep very close tabs on what is the label, which plant am I taking data on. Okay, so that's longest leaf length. Then the next metric we take is the number of green or chlorophyllous leaves. Now that can be really tricky on a rosette plant because how they spiral down, 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 down. Like how we could count down how many total little leaves there are. It's really hard to do without like tearing this whole thing apart. And there's so many little ones deep down in there. So we decided we needed a place to stop counting the earliest or youngest leaves that is the same sort of metric stop point across individuals, across researchers. So what we decided to do was identify the youngest leaf that is at least 75% of the longest leaf length at that time point. So we've actually done this on calculator. I don't have that ability in my brain, but I can tell you that 75% of 170 is 127 0.5-esque, 0.5. Okay, so same tool. Precision in math is important. So 127.5, doing the best I can. Again, I'm lining it up. 127.5, new pinch mark. Back to my same individual, and I'm looking at leaves younger or more to the inside, often lighter in color than that. So you have to really look at the order in which they were built, how they're spiraling and nesting on each other, and go kind of in order. So looking to the interior, here is a bright green leaf, and it is longer than that pinch point. So shorter than I wouldn't do. So okay, that's good. You're really close to that. I'm gonna come to an even younger leaf. And again, I'm looking at what is nested inside of it. This leaf here is the next youngest one. I bet you can guess. You think that's gonna be the length I want? Nope, it's too short, okay? So then I just go back to the one that was slightly longer than, but not shorter than. I'm like, okay, you are my very special 75% of this young leaf. So this is essentially the stopping point of counting. Okay, in fact, I'm gonna remove this one so we're not distracted by which one. So how many green leaves are there? Well, here's the thing about bromeliads, right? Some of the oldest leaves, which are furthest down or to the outside, they senesce or die and fall apart. Some of them readily just fall off, like these, and some are in the process of dying. So we need, again, sort of a cutoff of, am I a leaf? Am I a leaf? Etc., etc. So we're saying any green equals leaf. So sometimes it's nice to do like a little prune on your friends as you're doing this. Starting at the bottom, I'll go back to the one I was doing here. So I have some dying leaves, but they still have some green and they are attached. So this is a way to do this and keep track of the leaves as you go without hurting them, writing on them with a Sharpie, anything like that. I kind of gently fold them down. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and then the last one I count is that one we identified, 14. So then I would say this individual has 14 chlorophyllous leaves on this date. It's sometimes a fun thing to do with a partner and see if you both come up with the same number. It's a hard one. Potentially you could take a photo and like mark them off from that, but a leaf might be behind another leaf and you might not be able to see if it's green at the base. So it's often just best to do it in real time together. One thing to note, right, is if I were to come back and do these plants again in 2 weeks, 2 months, 6 months, whatever, whenever your interval is, which individual leaf is the longest, and then which individual leaf marks that 75% cutoff, it's probably going to change, right? So it may not make sense for me to continue to have a label on an individual leaf because the identity of what I'm counting is going to change over time. So like with this friend here, right, some of these younger leaves might get much bigger than some of the leaves right now that I might consider the longest, etc., etc. So you really have to kind of start from scratch each time you do these measurements, not just track the same leaves over time. The final thing I would want you to do when you're taking data is to check in on these meristems. So check the shoot apical meristem of each rosette and sort of categorize it. So in this case, for this individual, all I see is water and leaves, leaves, leaves down to there. And I don't need to stress too much about really looking down in there because I can tell you from personal experience, it's very dramatic and fast when it starts to build an inflorescence. So this individual, I would say, is vegetative growth only. I might write down those words, or I might say the category 0 vegetative growth only for like this individual. In contrast, right, both of these individuals, the shoot apical meristem has now produced an inflorescence. I may want to talk about categories about, are the flowers open? Are there fruits? These are young inflorescences with no open flowers. Maybe that's like a category 1. So I could maybe have a column on shoot apical meristem, 0, 1, 1 on these different individuals. Finally, I would want you to check for axillary ramets, or the little pups. And so you're going to want to peek often on the lowest or the oldest leaves and count them. And here's 1, here's 2, here's a little one, 3, here's 4, here's 5, even on this date and time. Then I can look up a leaf and I see nothing in here. So again, you're kind of going in developmental order. And in this case, there was 5. This individual, I see 1. And I see 2. And again, it's so big and dramatic, right? That don't stress about like getting in there too much or pulling it apart. That's going to cause more damage to the plant. Production of these, as well as production of the inflorescence, happens fast and big in these plants. So you're going to see it if it's there. So those are great data to take periodically, and lots of people can get involved in this type of data and comparing it over time to tell all kinds of exciting stories about bromeliad biology. So thanks so much.